Yeah, welcome everybody to the California Botanical Society seminar series. Thank you all for being here. Before I get going, I just want to mention a couple of things. If you can all, I see everybody seems to be muted. If you can just keep yourself muted during the talk, that would be great. And then we'll have questions at the end. I've also just set the chat up so you can just chat with the hosts and co-host, which is Glenn. We did that because of some unfortunate stuff going on in the chat in a particular conference. So if you need to say something, it's basically going to go to me. Also, we're still looking for seminar speakers. We have a couple of slots left, I think, over the summer and in the fall. So if either you are an early career botanist or you know an early career botanist who is studying the flora of Western North America, please let us know. If you are finishing your PhD, just finished your PhD, doing your postdoc, finishing your postdoc, starting your whatever your career is, etc., let us know. We will be happy to hear what you have to say. Okay, so tonight's speaker is Glen Morrison. And I am not in any way unbiased about Glen because he was my PhD student. And I think very, very highly of him, in spite of the fact that according to his mother, he almost flunked out of high school because he was just skateboarding and not doing his schoolwork and things like that. But and then he did finish high school though, and then he went to Citrus College, enrolling in a program in audio engineering, but somewhere along the line, he took an ecology course and he thought, I think I like this better. And so he then switched to ecology and followed up at Cal Poly Pomona. And at Cal Poly Pomona, he was in the McNair Scholar Program, which is how I met him. Before he graduated college, he spent a summer in our lab, basically spent the entire summer trying to extract DNA from silica dried and other manzanita specimens with essentially no luck whatsoever. But he stuck with it and then even decided to follow up by joining us for his PhD program. And so he finished about a year ago, and he will tell you about his what he learned. But in the meantime, as many of you know, I like to share with people something about our speaker that doesn't have anything to do with botany, something that they enjoy doing, some kind of a hobby. And Glenn enjoys woodworking. And so I want to share with you a picture of this. Can you see the picture? Yes, can you see it? Yeah. So this is Glenn and his wonderful wife Jazz at their wedding, and Glenn made this arch-like this
hexagon, yeah,
hexagonal structure behind them. So that's an example of his woodworking. So okay, so with that, I will turn it over to Glenn. So share your screen and let's hit the road. Oh, and yes, again, anybody who's unmuted, if you can please mute yourself.
All right, thank you so much for the great introduction, Amy. I'm really excited to give the talk to this excellent group. It's nice when you give a talk to people who are like really botanists, really know like get why plants are interesting, and this is a perfect such group. So this is a very boring title that I've landed on to describe the set of variously kind of tangentially related projects I worked on through my dissertation. Generally all have to do with things like patterns of morphological genetic diversity in the manzanitas, and you'll see how they tie together as we go. So to jump right in, if I can get my screen to there you go. So hopefully everyone knows what manzanita is. If you don't, this is a special treat for you. The Latin name for these guys is Arctostaphylos, which Amy knows I tend to say instead of the easier to say common name. And what's really interesting about these plants, other than them being just really highly charismatic, and I'll show you why that is that they are actually the most species diverse woody genus in all of Western North America. And that's saying something, because we're talking about a biodiversity hotspot. It's really off the charts. And this is the woody genus that has the most species by like a factor of two, too. So this is an incredibly diverse group. What they're probably best known for, including among people who don't care about plants generally, is a really like remarkably beautiful red, smooth bark. This captures people's attention who are completely plant blind otherwise, and is definitely the most intriguing thing about them to most people. They have a variety of different growth forms. So some of them are actually really, really low, like things you could easily walk over, like a ground cover, very prostrate little perennials like this. And you get like lower shrubs as well. This would be Arctostaphylos patula up in Convict Lake, which is just a really beautiful location. This is a plant here, maybe a third of a meter tall, next to a person here for scale. And then sometimes you get really, really large ones too. I'm going to show a stupidly large manzanita, just to be frank, next to the same person for scale here. So this is like the tallest one I've ever seen with my own eyes, in my life. But obviously not like gigantic trees, right? But you can get to where they're almost trees themselves. But while I'm highlighting this diversity, I don't want to oversell like how remarkably different they all are necessarily, because in a lot of ways they're actually quite similar, and that's one of the things that makes them a little frustrating. As I dig into this, and really most are shrubs. Almost all manzanita species are shrubs, meter to 3 meters tall or something like that. What you're thinking of when you think of shrubs, and the differences, unless you've really spent a lot of time looking at the differences and learning to spot them, and you've seen all the variability, they could be kind of hard to notice among a lot of species, at least. So, sort of as an example here I have 4 photos of one species. I'm going to do a little compare contrast with photos of the next species. So, we have kind of pointy leaves here, simple elliptical shapes, kind of, generic green color foliage, right? Let's compare that with these pictures of another species here. You can see there's some differences, right? But there's a lot of similarities here, right? Although I did also just lie to you, and this is 4 different species here actually that are just super similar looking. And I know people like Tito in the group who could see all the small differences, like those don't look similar, but I think most of us probably who haven't spent a lot of time looking at manzanita morphology probably would not have noticed the differences, right? And likewise, this is for other species that are just remarkably similar, unless you know what you're kind of looking for when you're interpreting their morphologies. And because of this, they're sort of like an emblematically, notoriously hard to identify group, and that's given rise to things like Tom Parker is excellent contribution of the field guide dedicated specifically to manzanitas, which sounds kind of crazy, right? A field guide for one genus, but it's not. In this group, it's so handy to have. Plug for Tom's book. I know he's watching right now, so pick it up if you don't have it. And things like workshops devoted specifically to identification skills for manzanitas. These seem kind of crazy, right? But anyone who's spent a sizable amount of time trying to start the process of identifying manzanitas will know it can be really difficult. And then kind of breaking this up a little bit more, more than just saying, they could be similar, right? To break this up into components that going to get into the studies I conducted. First of all, the traits that are used to differentiate the manzanitas can be really hard to understand on a fundamental level, like just to get an understanding of what is being described can be difficult. And then even when you have the understanding, the scale of the differences, I should say, are often rather slight between different taxa, and it can make it really hard to tell what you're looking at. And then and this is more kind of systematic of a frustration, but you often need reproductive stages that are kind of like out of sync in terms of one visit. You might need to make multiple visits to a plant or hope for old bits of something to be hanging on to be able to actually get at multiple phenological stages. So in terms of those hard to understand traits that are used for descriptions, I think hairs are the best. In manzanitas, you're often looking at whether you have glandular hairs or non-glandular hairs. You're looking at the density of hairs, and you're looking at like the overall average length of hairs. So here are 4 close-up photos of 4 different types of hair morphologies, ranging from no hairs, my favorite type of hairs because they're the easiest to understand, to things like sparsely dense dense, right? So not very dense, long non-glandular hairs to dense, medium length glandular hairs, right? And you can see how this could be really, really difficult for someone who hasn't seen all of the range of variability to kind of intuitively understand. So this all feeds into why they can be so hard to identify, as well as I pointed out before, sometimes you need different parts, different organs from different times of the year, effectively different phenologically exclusive times. So we call them nascent inflorescences in manzanitas. Basically, these are like the inflorescence sits around is formed and sits around kind of inactive for a number of months before it starts actually producing flowers. And the variability in those is viewed as being very diagnostically useful. But then often, almost always, you also still need fruit to identify them. And basic botany, you might know that it's a little bit difficult to have fruit and the thing that gives rise to the thing that gives rise to fruit at the same time. So these can be out of sync when you visit the plant for one time, right? Okay, and then when we start looking at morphological differences among subspecies, things can get even more murky, which is perhaps not a surprising statement to make, right? But to give a specific example here, if we look at Arctostaphylos glandulosa eastwood manzanita, there's 10 recognized subspecies of this plant currently, and a lot of the ranges of these are actually highly overlapping. And in some places, like this little inset map I have here of San Diego County, this is rough perimeters of the ranges for the subspecies down there. And in some places, you can find multiple subspecies in the same populations. In the most extreme cases, you can even get like 5 identifiable subspecies in a single population. And like I think I don't probably have to explain anymore why that would be really hard to deal with. It'd be a lot of overlap. Another complication that could be afoot, making all of this a lot more complicated biologically, and as it actually applies, is that hybridization has been documented in the manzanitas. There's been several studies over the course of several decades that have shown that it does happen, and it's suspected to be rather frequent. So probably a lot more common than has been already documented. And obviously, if you have hybrid individuals in the mix, that means you're going to have a potentially confused pattern of morphology. You don't know what you're necessarily looking at, species A or B, right? If there's a XB in the mix. Another thing that also complicates this pattern, this is a little bit less obvious, but I'll explain why this is that there's like a majority of species in the genus that are narrow endemic species. So endemic species to a very small little range. And that in itself isn't, would confuse anything. But what gets complicated is a lot of the time these narrow endemics the ranges of them happen to be rather close together, and the morphologies can be rather similar in little kind of geographic enclaves. And I'm going to give you a concrete example of this. So these are the what we call auriculate taxa of Santa Barbara and San Luis Obispo County. So these are rough range maps of the three Pichoensis, Purissima, and Refugiensis. And I'll just show you kind of like similar comparable parts of plants of each of the 3 species. And you could probably recognize there's a number of traits that make these noticeably different than other manzanitas for when they really don't have petioles. So the leaves are sessile, right on the stem for the most part, and they're highly imbricate, right? They're kind of like forming this scaled structure down the stem. And overall, they're highly similar. And of course, they're also over very small ranges, really close together. So this kind of begs some questions, more philosophically, like, would these perhaps fit a model of subspecies better, or populations even of the same species, right? So there's kind of levels of variability we're comparing across the taxa, and it makes it really kind of formidable for someone coming into the genus trying to understand it. I can attest as someone who did that. So that leads me to articulating my actual set of questions that I was approaching in my dissertation work here. The first one here is, to put it simply, how do we best divide the genus into species? How do we best do that actually conceptualize a species? And then, taking that from that level down, how do we divide species into subspecies? Really fundamental questions about understanding diversity of manzanitas. And another key question I'll be working towards, particularly towards the end of this talk, will be another really key question here. Like, as I said, the endemism of manzanitas in Western North America, really what's the kind of epicenter of their endemism, though, is just the California Floristic Province, which always ties my tongue, so I'll say CFP from now on. But the CFP, as we probably all know, right, is the majority of the land mass of California and a little bit of Baja, as well as Southern Oregon, that has that Mediterranean climate zone. And you actually find all 60 currently recognized species of manzanita here, and there's just a handful. There's like 4 or 5 or so that you'd find outside of this region, and all of them are otherwise represented here. So it's an obviously interesting pattern of endemism in this area. And there's really been 2 hypotheses put forward to explain this, and these aren't unique to manzanitas, obviously, and there's lots of patterns of endemism across the flora of the CFP. But the first one that's often sort of been taken for granted, I think, in the literature, or assumed to be true, is that this is really just a simple story of diversification within the CFP. There was ancestral species there, and they diversified like crazy. This is like an eminently reasonable assumption to make, right? A very reasonable hypothesis to put forward to understand the diversity we see, right? But it's also important to keep in mind that Western North America, even though outside of the Mediterranean climate zone now, is very dry and kind of inhospitable outside of the higher mountain ranges for manzanitas. It didn't used to be so arid and hot, right? We used to have much more temperate climate across much more of Western North America, and the flora was quite different. But in the fossil record, we know this, right? And we do know we have fossils of manzanitas over a much wider area. Getting into species gets a little more complicated with that. But this suggests that perhaps some of the diversity that now exists today didn't necessarily originate within the CFP. And I want to highlight, of course, both of these can be true, right? It's entirely possible that there could have been extensive radiations going on in the CFP, as well as radiations going on outside of the CFP, and then being preserved after they dispersed into it, right? It's not either or, these could both be contributing. So putting that all together, right, question 1, as I will be referring to it through the rest of the talk, is how do we best describe manzanita species? Then 2 is how do we best divide those up into subspecies? And then basically, what are the geographic origins of manzanita diversity as we see it today? So getting into that first question here, I'm going to be going through the results of a morphological analysis that is really still ongoing. We're currently adding some data to it. If you're curious about that, I could talk about it at the end. I don't want to burn through all of my time though, so I'll leave it. I'll just get into my questions here. So first of all, these are going to seem kind of similar, but I'll explain why they're different. The first question is, are manzanita species and subspecies distinguishable? Can they be told apart? And then kind of related question, but a little bit different philosophically, is how does the natural kind of spread of variation morphologically correspond to the taxonomy, right? And I'm saying distinguishable and distinct, and you might think why would you even? That's the same question, right? But I'm going to tell a little story here to make my point. So let's say you have a friend that has 2 little girl twin daughters, right? And you can't tell them apart for the life of you, right? But your friend seems to be able to tell them apart just fine. You think, well, certainly they can see something. There's some freckle or something I'm missing that they can see. And you ask, how do you tell them apart? And they say, oh, well, every day we dress Alice in green, and every day we dress Debbie in yellow. So, and this would be a case, right, where the twins wouldn't necessarily be like, it sounds weird to say about humans, but morphologically distinct, right? You wouldn't necessarily be able to tell them apart otherwise, except for you have this clue, right? That's been enforced by the parents that allows them to be distinguished. So that's what I mean when I say distinguishable versus distinct. Or taxonomy would be the equivalent of the wardrobe here, right? How we identify certain things to allow things to be told apart. Okay, so getting into methods here, I included currently we're up to 43 taxa for this. This includes a variety of subspecies as well as species level taxa. And we have a myriad of variables we measured. This really took like a lot of manpower and time to do this project. It was quite a lot. We had 32 variables in total. This included aspects of leaf shape, right? Hair length and density in different variables, plant height, dimension, that kind of thing. Many more I'm not going to list out because they would be both hard to explain and it would take too long. Currently, these analyses have no reproductive traits. I am looking into adding nascent inflorescence measurements. So that will change over time if we get all that data squared away. In total, this is about 350 herbarium specimens. And for narrow endemic species, right, which really kind of only tend to be in like one population or what we might reasonably recognize as one population, that could be as few as 5 samples for that species, trying to keep things manageable with how much variables we're measuring. And then for more widespread species taxa, we were looking at least 20 to 25, so many more specimens for things that are more widespread we might expect to be more variable. Okay, and then getting into actual methods of data analysis. So getting it to this question, are Manzanita species and subspecies distinguishable? This really lent itself perfectly to a method of analysis called linear discriminant analysis, or you may have heard LDA before. It's a very early machine learning method, technically speaking, not as interesting as the stuff that's going on now, obviously. But this basically allows you to take a set of variables along with a set of labels for what group it's supposed to be in, right? So even from the very first time this was published, it was applied to a species limitation kind of question. And so basically what we're doing is we use all the data along with the species labels to train an LDA model, right? Like come up with this mathematical model of how to tell them apart based on the variation. And then the reason this is informative is that you can actually run kind of random simulations on that model, and you can evaluate how well it does, how accurate it is at classifying things at different groups. So like if there's no pattern at all, if there's no signal, no distinguishability of the taxa, it would have effectively no accuracy. And if there is actual signal, i.e. distinguishability of the taxa, right, then you get high predictive accuracy. That's how you effectively apply these. And then I used another method. This is going to sound like Star Trek talk. As much as I love Trek, this is not as complicated as it sounds, but it's non-metric multidimensional scaling, NMDS, and hypervolume overlap calculations. This is a lot. So what I want you to look at, understand this as, is we're basically asking the question with the number of complicated statistical procedures. How much does species X overlap morphologically with species Y? That's really all it is. But because we have so many variables, we have to go through a number of relatively complicated mathematics to get to that simple understanding there. So that's all it means, even though it sounds like I'm pranking you with the description there. So I'm going to give you a basic kind of breakdown of how the graphs you're about to see are should be interpreted. I'm going to give you specifically one for LDA, but the overlap analyses have a very similar interpretation that I'll explain a little bit more when I get to it. So let's say on here, it's a tabular format. And let's see here on the rows of the table, we have a human determination, right? Or human expert determination of what it should be based on running it through a dichotomous key, something like that. And then on the top, we have the LDA prediction. So we're basically we're training this model, we're running it, and we're seeing its accuracy, right? We're going to do that many times. And what that allows us to do is to then understand how accurate it is, right? So to go back one slide, I've bolded out these cells here on the diagonal because that basically represents accurate classification. If we thought it was A and their signal, right, and LDA could pick it up, it also placed it as A, right? But there's going to be some range of variability, right, in terms of accuracy. So we're going to use a color scale to show that. So let's imagine a scenario with just a very kind of toy example with just 4 taxa here. So in this case, right, our human determination it was species A. We fed our samples in, trained the model, and we're getting the accuracy out. And in this case, A was 100% classified as A, no misclassifications. And then B, maybe we have relatively high, maybe not like super high, but high, like pretty good classification accuracy, but not perfect, right? And then maybe in this scenario, it's almost like a coin toss between the accurate species, the thing we thought it was, and something else that's perhaps similar morphologically. And then in a kind of extreme scenario, there might be like no signal whatsoever, really, no differences between this one and any other. So you just end up with like equal probability of it being assigned. That's the kind of intuitive logic that you should apply to the following graphs because they're going to get larger and harder to understand. But that's what you should be looking at. So to jump right into one of those, this is LDA trained specifically on species IDs. So you'll notice here, all of these are just species level identifications. And you'll notice first and foremost that diagonal is really easy to spot. And it's a really deep color on our color scale because this basically had really high accuracy. The LDA trained on species revealed very strong accuracy, suggesting that at the species level, at least, we have some like a decent level of distinguishability of the taxa, which is an exciting result, right? Any square here that doesn't have a number was 100%. Any that has a number is a lower percentage. You'll notice there's just kind of a few noticeable departures from that diagonal overall. And then when we include subspecies, things get a little more interesting, a little bit more chaotic here. And probably your eye is drawn to this area here. And that area there is actually all of the glandulosa subspecies that I included in the analysis. So remember, I mentioned that as an example of one where subspecies differences could be a little bit hard to tell, ranges overlapping, that kind of complicated scenario. And we see that pan out in terms of like the statistical measure of distinguishability based on the morphology. And this is perhaps to be expected, right? We wouldn't necessarily expect subspecies to be just as morphologically distinct as species. Another thing I'm interested in, if I will eventually finish writing and publishing, is kind of like a paper on the subspecies problem. If you extend the subspecies or the species concept problem, this is very much unsettled philosophical territory, but this is panning out in the data is what I mean to say here. And then moving on to the hypervolume overlap data here. Remember, this is just, you're asking how much does species X, right? So let's say this is species X on the rows of this table overlap morphologically with species Y, i.e. something on the columns here, right? So this is a unidirectional comparison. That's how you would translate this. How much does Andersonii, for example, overlap with pungens, for example? That's how you read this. Put it on a simple white, no overlap whatsoever, deepest blue, entire overlap, and you'll notice the diagonals here. But this isn't a result really so much as just articulating by definition, species A overlaps perfectly with species A, of course, no duh, right? And it's really where you start getting colors, in the cells off of that diagonal where we're getting some informative results. So I'm going to highlight here. What's really interesting is that some species or subspecies are overlapped by multiple other species. So, and I'm saying that on purpose, are overlapped by, right? Because if you look at Manzanita subspecies Manzanita here, it doesn't actually overlap a lot of other species and subspecies substantially. But if you look at all of the other ones that overlap it, right, it's basically suggesting that some species have a lot of variability. They kind of occupy a large morphological space, if you will, and other species are, very much overlapping in that space. And then there's actually in this particular analysis, I actually only had one taxon, one species in particular, Nissanana, that had zero overlap with anything else. Lots of them had relatively small numbers, but only one actually had zero overlap, interestingly. And then I'm going to highlight again the glandulosa subspecies, which very similar to the result in LDA analysis, show a lot of overlap among each other, as well as overlap with some other things as well. And then make this quick, but breaking this down in terms of narrow endemic species and widespread species, interestingly, I found that the narrow endemic species only kind of slightly overlap each other overall. There's not a lot of overlap between them. Interestingly, even though in some cases they can seem kind of hard to tell apart, at least at first glance with something like those auriculate species in Santa Barbara and Slough. But on this chart, it's actually showing there's not as much morphological overlap as you might think. However, all of the species, including the narrow endemics, tend to overlap the widespread ones much, much more. That's what's being shown here in this result. The pink colored ones are widespread species, using a kind of somewhat arbitrary classification of calling them narrow or widespread, rather than a quantitative one. But it's a pattern that seems to shake out in this data. And highlighting here, for example, glandulosa as a whole, rather than being split into subspecies, right, is many species are overlapping with glandulosa, as well as with manzanita, subspecies manzanita, again, similar to that last chart with the LDA, or sorry, with the first overlap analysis. Okay. So looking back at our questions, are manzanita species and subspecies distinguishable? I'd say the answer, a pretty large, to put it simply, is yes, but it does seem that pattern is driven mostly by specific diagnostic traits rather than like an entire lack of morphological overlap, because there definitely is some morphological overlap between many species. And then getting into our other subsidiary question, which is, asking about distinctiveness, right, rather than distinguishability. This took me to another kind of tried and true statistical method that we call k-means clustering. So this is basically a statistical procedure where you have, some table of many variables, right, and then you're going to you have sample of them, and you're going to basically specify a number of k, right? 1 or 2 or 3. You go through the procedure trying different numbers of clusters you're going to try to sort them into, and this algorithm basically will try to place each sample into one of k clusters. So it's pretty straightforward and widely used method for doing this kind of clustering, where you're just looking at the overall range of natural variability and seeing where the natural clusters are, to make it simple. And then you go ahead and you compare the clusters with species or subspecies, and if those are the same, right, all the samples from species A are all in k cluster 12 or something, right, then there's a real natural pattern there. And what I found basically was the k means result was super duper unstable. And this isn't like a failure of the statistical model. This is to say that there isn't like a natural kind of paradigm to divide, at least based on the morphological data that I had. And there isn't a clear natural way to divide these into really any number of straightforward clusters. And what I'm showing here actually, so this is, I ran the procedure, the k-means clustering procedure, 1,000 times, randomizing as I go. It includes a randomization function when it's making these classifications. And this is just the number of samples in the largest cluster, right? So when I ran 1,000 times, you'll see there's like in one scenario, it found, 36 samples in the largest cluster, and another, closer to 15. So what this really shows very clearly is that it wasn't finding a clear and stable solution to the clustering, which is in itself actually a result that is biologically interpretable for us, right? It's basically saying, when it comes to question 2, it appears not that the taxonomy doesn't neatly correspond to some pattern of natural morphological clusters. Okay, so then moving into our second question here, how do we best divide species into subspecies? This is not going to be a morphological analysis, although we've kind of touched on the question in relation to that in the previous section here. But I'm going to outline the results of a genetic study that we did here, particularly focusing on the subspecies of Arctostaphylos glandulosa. So just to review, there's 10 accepted subspecies. Most of them are found only in Southern California. There's a few that get further north, but most of the diversity in subspecies is down here in SoCal, and they're classically difficult to identify and distinguish from each other. And as I said before, you find many subspecies, sometimes multiple subspecies in the same location. So this is like we kind of went into this one as like our first major project in the lab working on Manzanitas, thinking like, if we could deal with this hairball can of worms, we could deal with anything, right? And interestingly, 2 subspecies of glandulosa are actually considered to be a conservation concern. One's actually federally listed, Crassifolia. So this is the results of the study that we published back in 2020. And basically, to summarize, we had 140 samples representing not all of the subspecies, but 8 of the subspecies. Pretty good taxonomic coverage, I think, though. And what we did is we generated genome-wide sequence data using a method called dd-radseq. I could tell you about if you're interested in the end. Going to simplify the technical details for questions if you like later. And then I analyzed genetic differentiation among the subspecies. Me and my Glen Yi-Huang, we're co-first authors on this paper. And I'm going to barrel right into the results here. But there's obviously nothing plotted on this graph, but I want to set it up before I actually show you the results so you understand. So this is a 2-dimensional plot of genetic space, if you will. This is a method called multidimensional scaling, similar to what I was using with the morphological data. Or very similar to something like a PCA, if you're more familiar with those as well. So this is basically a way of plotting genetic difference in two dimensions, even though we're working with a much higher dimension data set containing thousands of alleles, right? And we're going to be looking at individual points will represent individual samples, and they're going to be colored based on their determination. So I'm just going to plot that up here, and what you will first notice is that there's this kind of big clusterfuck here of different, largely overlapping clusters. Not every single one is perfectly overlapping every other one, right? But where we have a bunch of these subspecies that overlap each other genetically, to put it simply. And we really only have one subspecies that would be subspecies gabrielensis here, San Gabriel Mountain manzanita. That's really kind of in its own unique part of this genetic space, to put it simply, which is a pretty interesting result. And I should point out, right, actually, that the federally listed subspecies crassifolia is actually very much in the mix genetically with other samples. And I will say also that other members of the lab are looking more closely at crassifolia and some other subspecies of manzanita. So we might get more data and more resolution on this as things develop. But as it stands with this, or at least at the currently published result that we have, it really is not a story of neat correspondence between genetic differentiation and clusters, or clusters of genetic differentiation in the taxonomy. And this is another way of looking at it. This is a structure analysis. So, and if you look at any individual vertical bar here, right? So like I'm going to highlight one here, pointing it out with the pointer. Hopefully you guys can see that actually. That represents one sample in our analyses, right? And I'm changing the number of clusters here. So very similar to the k-means clustering result. I'm saying 2 clusters here, and then now sort them into 3 clusters, now sort them into 4, right? And after that, we really just, there was no reasonable pattern to be derived. So I'm only showing 2, 3, and 4 here. But you'll notice in some cases there, like for instance with gabrielensis, there is proportionally more of one cluster assignment. And I should say also, these colors are totally arbitrary. They're just to represent the different cluster assignments. It's not on a scale or anything like that. And you could recognize in some cases we do have, like gabrielensis has more assignment to this cluster I'm coloring yellow here, right? But it's not unique. Like for example, Cushiniana also has some of that as well, right? So overall, it's not corresponding to the taxonomy. But interestingly, if we take the same set of samples and the same cluster results, right? I'm showing here k equals 2. It's the statistically most favored result, so that's why I'm showing it here. But also because I think that it's showing something geographically interesting. And what I've done here is I've sorted the samples by latitude. So not by distance from each other or anything like that, but just simple latitude of collection. And you can see there's a really obvious pattern. There's a geographic kind of genetic cline going on here. So the model of the more obvious pattern of genetic differentiation in glandulosa is not in correspondence with the subspecies, but really in terms of geography is the simple way to put it. Okay, and then flipping around here, it's a little delay. Yeah. So to get to my last question here, when asking basically what the geographic origins of manzanita diversity are, it's going to be a little bit of a shift in methods and questions here, right? But first, I'm going to kind of lay the groundwork so you can understand a little bit how I came to this project and a little bit more about the kind of results when it comes to it. But first of all, it's focusing on these 3 species here. I'm focusing on Patula, Pringlei, and Pungens, very annoyingly all with P's because there's lots of P taxa and manzanitas. And these are pretty widespread manzanita taxa. So you get Patula through the Sierra Nevada and then like up into the Colorado basin here in the Utah and parts of Colorado itself. And then a little further south, you get also widespread species, Pringlei, and then stretching way down to literally down to Oaxaca state in Mexico, you get Mexican manzanita or pungens, very widespread and very much outside of the CFP. And I thought these would be really excellent species to look at, to get samples of across the range and try to figure out, if we can, using genetic information, what the most likely point of origin or what their geographic history is, right? So dive into that. And to remind you, the kind of 2 hypotheses have been put forward here are for overall diversity, for the endemic diversity of manzanitas in CFP, right? Is this kind of dichotomy of diversification within CFP versus species evolving outside of it and being retained in the CFP as they went extinct elsewhere where they might have evolved, right? To put that in mind. And to quickly run through the methods here, we collected samples, me and my collaborators here. This is a multi-person effort, multinational effort to collect samples across Mexico and across the United States, in the Western United States. And we tried to sample as broadly as possible across the ranges of these 3 species. That really was enabled massively by international collections. Would have been very difficult. So I really am very appreciative of my international collaborators for this, as well as Angela Bielman here from UCR, who helped me immensely in the Western interior of the US. And what we did is we collected as many samples as we could. We ended up preparing DNA libraries, dd-radseq libraries, for about 238 samples. Of those, we didn't have the best success or in terms of our sequencing result. Unfortunately, things happen with it. Unfortunately, a little bit, you know, the gods were not in our favor there, unfortunately. But about half of them did produce pretty good, usable, satisfactory data. So from there, what I did was I processed our raw data using the IPyRad pipeline, which many of you have probably heard of. It's a very standard way to do that. Then I eliminated admixed individuals using a structure analysis. I'm not going to have time to actually present like where I found those and so forth. If you're curious about it, I could tell you about it. But I did find admixed, presumably hybrid individuals in several of these species, or that would be identified as several of these species. So that's in itself an interesting result. But using the ones that did not appear to be admixed, I built a phylogeny, a basic time-calibrated phylogeny. And then what I did was I used a model or a statistical package called biogeobears that basically can take geographic information into account and model the geographic history, the biogeographic history of a phylogeny on top of it. So we were able to come up with hypotheses, favorite hypotheses, as to whether as to where the species originated and how they spread out over space. So, and I'm going to give you a very quick primer, geographic primer, because I'm going to be using the names of these mountain ranges. So obviously everyone should hopefully know we're here in the California, what the CFP is. I don't know where you're zooming in from actually, but that's their area here, California mostly. And then I'm going to call this area SoCal, not too hard to understand, not necessarily a geographic kind of entity, but not like as textbook as the others, but I'm going to make it simple for our purposes and call it SoCal. And then we have the Basin and Range Province, which is all of the things like the Madrean Archipelago here, the higher ranges kind of in the desert Southwest. Then we have the Colorado Plateau, which is like, think Utah, parts of Colorado, obviously, as the name would suggest. And then we have the Sierra Madre Occidental, the Sierra Madre Oriental, west and east, obviously, if you know a little Spanish. And then we have the Trans-Mexican Volcanic Belt here, which runs east and west in lower Mexico. And then we have the Sierra Madre del Sur here, so the southern Sierra Madre. Just for a little bit of ground rules, so you know what I'm talking about when I start talking about these. Okay, jumping straight into the results for Apatula. What you're going to want to look at is the region that is inferred right here at the root of this tree. This is the region that is favored as the ancestral region, right? And you can sit here and kind of pick apart what this tree is saying, but I went ahead and translated this into a kind of map format so you can make this a little easier to understand. So the southern Sierra Nevada here is inferred to be the ancestral area, or the most ancestral area represented in the data set. And then something like 9 million years ago, we had dispersal towards the northern end of the Sierra Nevada. And then around the same time, maybe 10 million years ago, there was dispersal from the southern Sierra Nevada into SoCal. And then subsequently, and much more recently, there was dispersal into the Colorado, or over to the Colorado Basin. So it favors a origin in the CFP. And then if we're looking at pungens, that one that's really extensively widespread in Mexico, it's not favoring one particular region, but two of the regions I defined, which would be the northern Sierra Madre Occidental, or the Basin and Range, as the most likely best supported ancestral kind of homeland for the species. And then this one gets really complicated to understand the tree, so I'm going to jump straight to the map again. So this whole area I've encircled here is inferred to be the area where the species likely originated, evolved, diversified, right? And then something like, about 10 million years ago, there was a dispersal into Southern California, which is the only place you really find it in real number in California, in the CFP. And then a little bit after that, around a similar time though, you had a kind of subsequent stepping stone history of dispersal southward into Mexico, or this is the favored model, of course. So something like 9 million years ago, we get a jump from the Sierra Madre Occidental down into the Trans-Mexican Volcanic Belt. And then after that, subsequently, there was dispersal up north into the Sierra Madre Oriental, and then most recently dispersal further south into the Sierra Madre del Sur. So it's a kind of origin outside the CFP, heading south. Okay, and then this one's really easy to explain. This is the Arctostaphylos pringlei. So this really sort of has two big clusters of its range, one in SoCal, one in the Basin and Range. And the model really was not able to, there was no favored single origin point for this particular species. It had about equal probability of coming from either. And in correspondence with the author of BioGeoBears, the best interpretation of this is really that that there's a history of vicariance here. That's sort of the single best hypothesis to put forward given this result in the analysis, is that the species likely had a relatively larger and more contiguous range in geologically recent past, right before the deserts formed here, and there is evidence for a kind of suitable climate in this region before, and then effectively it got divided when the deserts formed, so it's most likely a vicariance event. Okay, so to kind of put those in perspective, we have one species in terms of the perspective of, the pattern of overall manzanita endemic diversity in the CFP, right? One likely originated in the CFP, one likely originated outside the CFP, and one has an unclear origin. So to put it simply, things may be a little more complicated in terms of the geographic context of diversification of manzanitas, right? But at the same time, I'm like literally finding evidence for kind of all of the possibilities. So I think it's an interesting result, but also, not as clear as one might hope it would be, right? Okay. With that, I want to acknowledge all of the many, many people who really substantially enabled this work, including Amy, of course, who's here, and other colleagues of mine like Tito and Tom that I know happen to be here, and anyone else. Thank you. I could take any questions.
Okay, yeah, let me change the chat settings so you can put questions in the chat, but otherwise you can just unmute yourself and ask. Andy?
As usual, I have a couple of questions.
Mhm.
First off, when you did your biogeobears analysis, how did you calibrate those ages of
Yeah,
those?
that's a great question. So, we have an estimate for the age of these. So there's an out group that I'm not including in the visualizations for one. I had a sample sequenced of Arbutus manzanita, which is clearly not in the genus, right? So it provides a good point of divergence. And one of Tom's previous papers actually gave a estimate of the divergence time for Arbutus from Arctostaphylos. So that sort of set the bottom line time calibration. Although one thing that is a little unfortunate is that beyond that, right, in terms of when this clade of manzanita is diversified from this other one, or that kind of thing, it sort of depends on a little bit better of understanding of the phylogeny in the genus, and that's very much like an ongoing work. We do know some fundamentals about it, but in terms of good time estimates, they're a little bit limited. So it's really just calibrated down to the root, unfortunately, which is yeah.
If I may, I'd like to introduce two words of caution.
Mhm.
Which would one which skews your result in one direction and the other skews
Yeah.
your result in the other direction. So first word of caution. The divergence point between Arctostaphylos and Arbutus is going to be Eocene, because that's the origin of that's when we first encounter
Yeah.
Arbutus in the fossil record. The is clades that are undergoing rapid radiations, and it is clear that Arctostaphylos is doing that. Whether it's inside of the California Floristic Province or outside, it's clearly doing a rapid radiation. That seems to coincide with more rapid rates of molecular evolution. This is going to result in the molecular divergence states that you are using from the ancestral clade being older than the actual date.
Mm-hmm.
However, I can tell you from experience working with the Malii that if you have a lot of hybridization within a clade, which there is in Arctostaphylos,
it slows down
as
the
we
estimate.
know,
Yes.
that skews it in the opposite direction. So
Yeah, no, these are all really excellent things to keep in mind. For one, right, this is still as of yet especially in terms of the phylogeography project, somewhat preliminary, right? For instance, I was just using a very simple algorithm to do the time calibration. It's basically based on geometry. I want to do a more a molecular clock-based method. So that's something ideally, we're going to get the rest of the samples that didn't come through in the first pass. In Amy's lab right now, they're prioritizing other kind of backlog of sequencing library preps right now. But the long-term plan is to get that other 100 or so samples that weren't included, and then really go hog wild getting the best estimates as possible. But honestly, you make an excellent point, even with the better inference of the topology and the dating, right? It's still like a mountain of salt to be taken on the potential dates, right? And I think something eventually when I publish this is, I plan to emphasize perhaps the relative orders of dispersal events more so than dates, because I think dates can obviously be suspect,
Well,
right?
The reason why I'm zeroing in on the dates though was because it's something that is important, and I will drop this and then let Clint ask his question. There are fossils in the Finger Rock and Stewart Valley floras in Nevada that I'm sure you know about, and I'm sure you know the ages of Middle Miocene. And it would be really interesting.
They're not as old. They're not super old. Yeah.
It would be really interesting to be able to get a handle on, are those fossils likely to be members of species or within the crown clade of Arctostaphylos lives in California, or are they likely stem taxa? Because that also has bearing on your question of where did this radiation originate?
Yeah, absolutely. And it's something I thought about a lot. The older ones, unfortunately, are just fossil species that they are not identified, or at least considered currently to be extant lineages, or ones we could recognize easily. And then the fossils that are known to or thought to be current taxa extant lineages are like very much more recent ones that like would do us much good anyway. But I very much appreciate the comments. That is a real hairball of something that's been a fun challenge to work on when doing this kind of reconstruction. Thank you, Clint.
I'm curious about the overlapping subspecies of Arctostaphylos glandulosa and how you think they might have evolved, and do they occupy different habitats? And so you have, two different groups of at least two of overlapping subspecies. And then does that occur in other Arctostaphylos species elsewhere?
Yeah, so I'll kind of work backwards. So there are similar scenarios with very polytypic manzanita species, like crustacea is one that comes to mind. It's not as diverse, but it's pretty dang close. I forget the exact number of subspecies, but there's a good number. And in some cases, you get them overlapping geographically. And in some cases, they're not. It's a little bit more well-behaved, I would say, than glandulosa is. But you do that. Like the current model of understanding taxonomy recognizes other species that have a kind of similar range overlap kind of complication. And then kind of working backwards in your question, in terms of like the ecological context in which you find them, in some cases, certain subspecies of glandulosa are really thought to be tied, and observationally really do appear to follow this rule, right, of being like occurring only on one geologic substrate. For instance, like crassifolia is a great example of that, where you find it in this maritime chaparral where you have a kind of relatively sandy substrate. Tito, I see Tito grinning. Please correct me if I'm wrong. He knows much more about this. Tom also as well, but like that's
this is why I'm grinning.
Oh, okay, unrelated, but that would be an example of one that's held up as like kind of abiding by some rules of different ecology, right, and environment, as well as Cushingiana, a little further north at least, there seems to be some kind of ecological axes where you find either Cushingiana or you find the other species that you find everywhere, Glandulosa. They do seem to kind of partition ecologically. In terms of like studying it rigorously, I think that would something that'd be really interesting to look at. But then in terms of some other ones, it's very much less clear. Like that kind of, mess going on in the interiors of San Diego County, for instance. Like we literally have been to populations where we found 4 or 5 subspecies that are pretty easily, at least pretty confidently, we identified too. So in that case, the situation of ecological partitioning is a lot less clear, right? I think Amy's going to say something.
Yeah, I just wanted to jump in and say that's a great question that as Glenn was saying, we find these different subspecies like right next to each other, and in the same populations, you'll find a lot of things you can't identify to any subspecies.
Yeah, also.
So there's sort of continuous variation, and I'm sort of jumping the gun here a little bit, but it's that particular species, Glandulosa, is tetraploid. It's a tetraploid species, and it looks like some of these tetraploid species may have multiple origins also. So multiple hybridization events kind of leading to similar phenotypes. So it may be actually that this is kind of just a hybrid swarm of individuals rather than a coherent species that you can sort of divide into really truly distinct subspecies.
So
Yeah.
why wouldn't you classify that as just variation within one subspecies?
Well, it would be actually within one species, but we sort of haven't gotten to that point yet. We haven't gotten, and it may turn out to be taxonomically very complex. We haven't gotten to that point yet. All I can say is that the analyses, particularly that Tito is doing at the moment, are suggesting that some of these tetraploid species may have multiple origins. So.
Yeah, and there just may be like when you combine various diploid lineages and they form these tetraploids, you may get ecologically similar, morphologically similar sort of monsters out of that combination, Although honestly, I don't want to jump the gun on your jump the gun because I'm not fully up to date on.
Yeah, I mean we're a long way from really having sorted this out. It's just something that we see in the tree as a possibility, but we haven't really gotten to the point of being able to say anything definite about it yet. So that's a very adorable kitten, Tito. Let's go to Aiden next because he hasn't asked a question yet.
Hi, thanks for the talk, Glenn. That was really excellent work you presented.
Thank you.
I wanted to ask about your sense of the role of environmental variation in morphology in these species. Obviously, it would be really challenging to do a 43 species manzanita common garden, and that would be wild if you did that. But I'm just curious, like how much morphological variation you think could originate from environmental factors.
Yeah, totally. And like when I started researching manzanitas, I was really keen on the idea that environmental plasticity could it just seemed like, there's all these slight differences and these, small ranges, right, on interesting soils. And I thought, what would explain that is environmental plasticity, right? But interestingly, and this isn't like, this is a little bit anecdotal, a little bit, but what I think there's sort of an informal series of common garden experiments that's been going on, which is just all of the ex situ conservation of these species. And when you grow these species out of their original environmental context, you do get very similar morphologies. It's not like there's like they're unrecognizable or something. They do by and large look like what they're supposed to in the field, which I think is a sort of like a pretty substantial weighed in the favor of it not being at least like mostly dictated by environmental plasticity. And I would say that, and this wouldn't be anecdotal, but there is also like one of the most difficult papers to read ever like, or I don't want to say it's poorly written because I don't want to offend anyone if they go and look it up and happen to have been involved with it, but like a very thorough experiment someone did, an actual common garden experiment of some now no longer recognized varieties of Uva ursi, which is the really, really widespread one that you can actually find in like Eurasia as well as, you know, like Guatemala, Honduras, or Tom Parker can correct me. There's even in South America, there's a population of volcano, but like it's been found like all over
Guatemala.
the Guatemala. Thank you. Yes, and so it's a very widespread one, and it was at least at various points in time described with some level of subspecific taxonomy. And there was a paper in the 70s, I believe. I'm not remembering the author right now, but they did a common garden, and they changed like some environmental variables they hypothesized to be relevant, and they did see some shifting in terms of like the hair morphology, like the development of glands or not, but they didn't see like a very substantial effect of changing the environment. So I would say overall, I think that environmental effects are probably like a smaller slice of the pie overall. But it would be interesting to look at more rigorously, you know, because at this point it's like I would even, you know, it's sort of writing it off, you know, but especially in things like those three subspecies real close together in San Luis Obispo and, you know, Santa Barbara County, I would be really interested to if someone did like a common garden on those or something, like if you got pretty different morphologies, you know. I think it's still plausible.
Great, thank you so much.
Before I let Andy loose again, does anyone else have any other questions they want to ask? Going once? Going twice? Okay, you're up, Andy.
Second question is also related to your biogeographic study. There have been potentially a fair amount of phylogeographic change, both in terms of where the mountain ranges were and how high they were, in addition to the usual sorts of climate change that happens over the late Cenozoic.
Yeah.
Are you at all concerned that this might affect your analysis or interpretations?
Yeah, so I mean, like, I'm just obviously thinking off the top of my head. For instance, like the Nevada Plano, for instance, like was effectively like its own geographic. It basically turned into the Great Basin instead of, you know, being this big gap before it was, you know, this massive plateau, right? So that's something that, you know, I'm thinking about a lot in terms of the interpretation of how things could unfold, right? In terms of like explicitly modeling for it, I think it might be a little bit challenging. Not impossible, right? But like I'm thinking, at least in terms of the kind of timescales we're contending with, I think most of the existing the geology and mapping across, you know, geography, I think maintains relevance today. I would be interested to know if if there's any particular parts of those ranges, for instance, that are like more geologically young or recent than I'm perhaps thinking of. But the main one that comes to mind for me is Nevada Plano, and it still like factors in because it ends up being a gap instead of, you know, habitat, right? Today versus, you know, before.
I want to say there's some question as to how high the Sierra Nevada were when.
That's super interesting.
There's question about how high the Nevada Plano was too.
Yes, I'm aware of that. Yeah, that one that's interesting.
Higher, probably higher than. Actually, I'm not sure because Nevada has the highest average elevation of any state in the U.S. now.
I didn't realize that. That's kind of cool.
Yeah, it's Nevada is already high, but was it even higher? And if so, how much?
Yeah. And the other parts of like you know, even south of what we might traditionally think of as a Nevada Plano, like for instance, would parts of what is now like the Mojave,
New Mexico,
higher, because that would have made a lot more substantial area for something kind of interior chaparral.
I don't know. So one, I don't know.
Yeah,
but also as far as I know, as far as I'm aware, it's not entirely settled by geologists either.
Yeah, that's validating to hear you say that because when I was like trying to absorb all this stuff for particularly for that chapter of my dissertation, like it's one of these moments where you make a foray into another science, you realize, oh, there's this whole other science here, right, that I'm not an expert in, right? But I think at the end of the day, when we come to writing an actual publication for those results, right, when they're kind of in the best shape and updated as best as possible, I really want to bring in, like, I really am interested in finding like one or two, like kind of persons of expertise on the kind of the geologic climatic history of Western North America to try to bring in, right? Because it's like it's definitely an area where you can kind of oversimplify or kind of misunderstand things. Like as you're saying, if you're misunderstanding the actual history of it, right?
And an awful lot of the paleo climate history is informed by paleobotanical
Yeah.
floras.
So it's sort of a circular loop.
And the paleobotanical floras are desperately in need of revision.
Yes.
They were collected by Axelrod in the 60s and 70s.
There's not that many of them either. Like when I was digging, I mean, maybe
there's a fair number. There's a fair number. There's well, it's patchy. So there are a lot in Oregon, and there are a lot in Oregon and Idaho. There are a lot in there are a lot in Nevada.
Yeah.
There aren't a whole lot in California during that time period. There aren't a whole lot farther east, and there aren't a whole lot farther south that at least have been described.
So let me interrupt for a second. We're kind of getting into the weeds here a little bit.
Yeah.
This is a rabbit hole.
Yeah.
I think you're getting it.
I see another hand up, and also I just want, it is 6 minutes after 8. Anyone who, feels they've heard a seminar, you're welcome to leave. And those of you who still have questions, feel free to hang on. Glenn?
Elder Glen. Yeah. Glen Holstein.
Oh, yeah. Glen Holstein.
Glen and Glen. One and Glen.
Yeah. I was wondering if on those coastal endemics, there may be historical islands that may have isolated them in that region.
That's an interesting question. Yeah, like basically with a higher sea level history, would some of those kind of small clusters or ranges have been islands at the time? And honestly, I don't know off the top of my head. I do know, I am aware of the history of, for instance, the channel lines having been connected to mainland at various points in time when sea levels were much lower. But thinking about the other way in terms of areas along the coast that would have been islands previously is not something I've thought about it a lot. And that's something that's totally worth considering in terms of the kind of evolutionary history going on. Yeah, that's how I answer that. That is a very worthwhile thing to look into that I have literally never thought about.
Awesome.
Yeah, I don't know much off the history off the top of my head.
Let's just say, does anyone else have any questions at this point? Give you a few seconds. I want to give a shout out to Tito for sharing his kitten with us. I think we all benefited greatly from that. Audrey, you have a question?
Yeah, just real quick. I have a series of questions that I would love to email you. However, I can't easily find your current email address.
Yes, yeah, I'm a little bit. I'm currently teaching at Cal State Dominguez Hills on non-permanent contracts, so my emails are a little here and there. I'm just going to put my. I'm going to put into the. You send me a message on chat, and I'll send you back my personal email. It's probably the best way to get me right now.
Great, thank you.
Teaching email, yeah.
Anyone else? Going once? Going twice? Well, thank you very much, Glenn. That was some fast talking you did there. I I admit, I was a little worried at the beginning, but you got in under the wire, so.
I always know I go over, so I try to
Yeah.
speak faster than I naturally would.
Yeah, that was impressive. So anyway, thanks very much, and see you all next month.
Thank you. I'm gonna email it back Audrey here before you close
Okay.
it.
Oh, are you emailing her?
Yeah.
No.
Or I'm gonna put it in the chat. Sorry.
Yeah. So I'll.
Thank you, everybody.
I will hang on until you get that in the chat. Thank you all.
Bye.
Thanks, Mike. Good to see you.
Good to see you too.
Hi, Mike.
Hi.
Okay, Audrey, I emailed you my
Okay.
best, most
I'll let you
stable
guys go.
email.
Thanks.
Thanks for coming, Mike. I
Mhm.
didn't even see it was there until the end there. Audrey, you got that?
Yes, I got it. Thank you so much.
Okay, before I disappear into obscurity here.
All right. You guys
the
have a
sun
good
goes
night.
down. Okay, thanks, Glenn. That was
Thanks,
great.
Amy.
Thanks.
Bye.
Take care. Bye-bye.